Every single one of us has an origin story. But it doesn't start with our first words or our first steps. Nope, it starts way, way before that, on a totally microscopic scale. It's a story about an incredible journey, with impossible odds and some seriously intricate biological choreography. So today, we're going to trace that story right from a race against millions to the exact moment a single cell laid out the blueprint for an entire human being. And our story, it starts with a number that's almost too big to imagine.
300 million. Yeah, you heard that right. That's roughly how many sperm kick off this whole race. It's just an astronomical figure. But it's totally necessary because of how incredibly difficult the journey ahead is. I mean, the odds are stacked against them from the very beginning. It's a brutal but super effective evolutionary strategy to make sure that only the absolute strongest contender even has a chance.
And it really is an epic quest. You can think of it like a dangerous odyssey through the female reproductive tract, where only the strongest, and let's be honest, the luckiest, will even get close to the finish line.
Okay, so this diagram here basically maps out the racetrack. The sperm start out in the vagina and have to travel all the way through the cervix, across the entire uterus, and then into this one specific spot in the uterine tube called the ampulla. And that's where the prize is waiting, the oocyte, which is the scientific name for the female egg cell.
Just let that sink in for a second. From 300 million starters down to just a few hundred survivors. The whole trip takes somewhere between 2 and 7 hours. And while the sperm's own tail gives it some push, they get a major boost from these powerful uterine contractions that kind of pull them forward. So, in a way, it's a team effort.
But here's the thing. Just showing up isn't enough. The sperm that survive the journey aren't actually ready to fertilize the egg right away. They have to go through one last, super important transformation to get the power they need to break through the oocyte's defenses.
And that transformation is called capacitation. It's about a 7-hour conditioning process where a protective layer, basically a sugar protein coating, gets stripped off the sperm's head. It's kind of like a knight taking the cover off his lance right before the final joust. Without this step, fertilization just can't happen.
So picture the oocyte like a medieval castle, right? To get to it, the sperm has to get through 3 layers of defense. First, there's an outer wall of cells called the corona radiata. Then there's a really tough inner wall, the zona pellucida. And finally, if it makes it that far, it has to breach the cell's own membrane, the castle's last gate.
And this brings us to the biochemical key that unlocks it all. To get through that really tough second barrier, the zona pellucida, the sperm kicks off something called the acrosome reaction. It releases these powerful enzymes that literally digest a path right through the shell. It's a one-shot weapon. And it's what allows just one single sperm to finally reach the egg itself.
All right, now for the main event, the fusion of sperm and egg. We're going to break this down step by step because this is the exact moment a brand new, totally unique genetic story begins. So first up is the corona radiata, that outer cloud of cells you see there in diagram A. The sperm uses its powerful tail for propulsion to basically just muscle its way through. Relatively speaking, this is the easy part.
Now, the tough part. In diagram B, you can see the sperm's head making contact with the zona pellucida. This contact is the trigger. The acrosome reaction fires off, releasing those digestive enzymes that start drilling a tunnel through this hard protective shell.
And here it is. Steps C and D show the moment of fusion. The sperm's membrane becomes one with the oocytes, and that lets the sperm's head and tail just slip inside the egg's cytoplasm. And the instant this happens, the oocyte's membrane slams the door shut. It becomes totally impermeable to any other sperm. The race is officially over. We have a winner.
So, once it's inside, the genetic material from both parents gets organized. The sperm's nucleus swells up to form what's called the male pronucleus, and the oocyte's nucleus does the same, becoming the female pronucleus. Each one is carrying 23 chromosomes, exactly 1/2 of the genetic blueprint. And then, finally, the 2 pronuclei fuse together. They combine their genetic information and restore the normal number of 46 chromosomes. This new single cell, which we call the zygote, is the very first cell of a new human being. It's got a completely unique genetic identity.
But hey, the story is just getting started. This single cell is not going to stay single for long. The first week is just this period of unbelievably rapid division and transformation, and it's all happening while it's traveling down the uterine tube toward its new home. This whole process is called cleavage. It's just a series of super quick mitotic divisions.
One cell becomes two, two become four, four become eight, and on and on. And what's really fascinating is that the embryo doesn't actually get any bigger during this stage. The cells just get smaller and smaller, and there are more of them, all packed inside that protective zona pellucida.
So these diagrams here, they show the first few steps of that crazy multiplication. The single zygote divides into 2 cells, then 4, then 8. You get the idea. This chain reaction of division happens incredibly fast, and it sets the stage for the embryo's first big transformation.
By about the 3rd day, the embryo is a solid ball of 16 cells. This little structure is called a morula, and it gets its name because it kind of looks like a mulberry. Right now, it's still just this tiny, compact cluster of identical cells. But then, something pretty remarkable happens. Fluid starts to seep into the morula, and it creates a hollow cavity inside. This transforms it into what we call a blastocyst. So now we've got 2 very distinct parts, an outer cell layer, which will eventually form the placenta, and this clump of cells on the inside called the inner cell mass. This inner mass right here, that's what's going to become the embryo itself.
So with the blastocyst formed and all ready to implant in the uterus, the next major event happens in the 3rd week. And this is where that simple little inner cell mass organizes itself into the fundamental blueprint for an entire body. Honestly, it's one of the most critical events in all of development.
This process is known as gastrulation. It's this massive cellular reorganization where the embryo goes from being a simple 2-layered disc to a complex 3-layered disc. And these 3 layers, they are the foundation from which every single tissue and organ in the body will come from. What's happening here is a huge coordinated migration of cells. You have cells from the embryo's surface that literally dive inward and arrange themselves into 3 distinct sheets of tissue. We call these the primary germ layers: the outer ectoderm, the middle mesoderm, and the inner endoderm. This simple 3-layered structure is the fundamental blueprint for the entire body. So, what do these layers actually become? Well, their fates are pretty astonishing. The ectoderm on the outside, that forms our entire nervous system, our skin, hair, and nails. The mesoderm in the middle builds all of our muscles, our skeleton, our heart, and our blood vessels. And the endoderm on the inside creates the lining of our digestive tract, our lungs, our liver, and our pancreas. Literally every single part of you can be traced back to one of these 3 simple layers. So from a race of 300 million to a single cell, which then divides, travels, and completely transforms itself into the 3 foundational layers of a human body, it's a process that every one of us followed. And it leaves you with a pretty profound thought, doesn't it? What does it mean that the foundation for your most complex organs, your brain, your heart, was all laid down by a simple migration of cells just 3 weeks after you began? It's an absolutely incredible origin story to think about.